Hi, let's talk about the central nervous system, the peripheral nervous system, and their structures. So the brain, as you probably know, is the command center for instructions for the body. The human brain contains about 100 billion neurons, and each individual neuron could have hundreds of connections to other neurons. We have yet to mimic exactly these interconnected kind of structures and the ability to make decisions using artificial intelligence. There are some new imaging techniques, and I'm going to show you imaging techniques that can be used on living humans to determine where activity is in their brain. This is one, this image over here on the left is one that's used in, it can't be used on humans because it's ultimately fatal, but this is one that's in a mouse where they use fluorescent tags of all different colors to tag different brain proteins, and that way they can tell by the color of each individual neuron which proteins it's expressing. Each individual protein will be a different color. So that's a really powerful technique, but one we have to only use on animal models. So we know that there were nervous systems of some sort as far back as the Cambrian, because we had animals that were bilaterally symmetrical and motile, which implies that they had both motor control and they had sensory input about their surroundings. Both of those things we'll talk about in this chapter and the next chapter. The simplest nervous systems are found in the cnidarians and the ctenophores. It's just a simple nerve net of neurons that both can receive input and react to input. They don't have a centralized decision-making center and no brain. So like this hydra here, they just have a network of nerves that together can make decisions. And cnidarians do have directional swimming, and they do capture and kill and eat prey. So they are doing things with their nerve net, surprisingly complex things, even though they don't have a brain. Once there is bilateral symmetry, and the simplest kind of bilateral symmetry is found in flatworms and other invertebrates, we now can have a head and a tail, and the head is where we find increasing decision-making and larger neural structures and a concentration of neurons that is involved in decision-making, and that is the central nervous system and the brain. The peripheral nervous system, as animals become more advanced, generally is not decision-making, but the peripheral nervous system is receiving sensory input and the motor instructions that go out to the rest of the body from the central nervous system. Although there are exceptions to that. For example, in cephalopods, they do have seemingly a decentralized nervous system, even though they do have a brain. Each arm can make decisions, it appears. So as the nervous system is organized, there are also clusters of neurons outside of the brain, and we call these ganglia. And we have ganglia as well, and we'll talk about the spinal ganglia when we get to the motor section in the next chapter. But there are annelids and arthropods that, in addition to the brain making decisions, there are decisions also being made in the ganglia. And we also have ganglia in the central nervous system in the brain. In vertebrate brains, the ganglia are called nuclei, but they're essentially a cluster of neuronal cell bodies. That's what a ganglia is. Or one would be a ganglion. The plural is ganglia. Just like for nuclei, which is not the same as a cell nucleus. Nuclei in the brain, nucleus would be singular, nuclei would be plural. And things that are motile require a more advanced nervous system than things that are sessile. So things that don't move around as adults don't need a nervous system. In fact, they often lose their nervous system once they're out of their swimming larval stage. For example, tunicates. They have they're chordates. They have a nervous system that they use while they're a swimming larva to activate muscle movement. But once they become sessile, they lose almost their entire nervous system. So here we have some other, some invertebrates and one vertebrate here. So we have brains, we have ganglia, and both of these can be important components to both receiving sensory input and sending out information to be acted upon, mostly in the form of muscle movement or digestive activity, which would also be muscle movement, breathing, heart activity. All of those things are under the control of the nervous system. For vertebrates, and this is what we're going to focus the most time on, the central nervous system is the brain and the spinal cord. Everything else is the peripheral nervous system. So ganglia that are outside of the spinal cord and next to the spinal cord, the spinal ganglia, they're not part of the central nervous system, they're part of the peripheral nervous system. And there is specialization, regional specialization, and we'll talk about some examples of that. Besides neurons, the most important cells in the nervous system are the glial cells. The glial cells, there are several different types, are support cells, and they have a variety of functions. In the embryo, glial cells help neurons to grow in the right direction. They create tracks that neurons can grow along. Glial cells are really important in the brain and the central nervous system and in the peripheral nervous system. They form the blood-brain barrier. They line the ventricles where the spinal fluid is. They restrict substances getting in and out of the brain. They get rid of the metabolic debris of the neurons. Neurons generate a lot of metabolic debris, and the glial cells clean that up, clean up the mess. They also help defend neurons against invaders like viruses and bacteria. So here's just a few of them, and we'll talk in more detail about their function. So we have central nervous system here, CNS, all of this, and then peripheral nervous system. So in the peripheral nervous system, we have Schwann cells, which we already talked about in the last chapter, as helping to speed up action potentials along axons by confining the action potentials to the gaps between the Schwann cells. And so those are support cells. Microglia as well. And then we have in the central nervous system, and we'll talk a little bit more about each of these, ependable cells, astrocytes, oligodendrocytes, as well are support cells in the central nervous system. And they also, unlike neurons, can divide. Well, not Schwann cells, but most of these cells are capable of cell division, which is why when somebody gets a cancer in the central nervous system, brain cancer, it's often a glial cell that is the original cell type that became cancerous, because glial cells have the ability to divide and neurons don't. Neurons are terminally differentiated like muscle cells, and they cannot divide. To generate more neurons, we have to have an undifferentiated stem cell. Radial glial cells and astrocytes can both behave as those kind of stem cells. There is research into triggering those cells to generate new neurons in the case of damage of nerve cells. And these experiments are being done in tissue culture and in animals like rats and mice with some success. The issue is often not getting the cells to divide, but getting them to turn into the type of neuron and acquire the connections that are normally formed during embryonic stages. later in life. But it's very interesting kind of research, looking at things like repairing damage to the spinal cord in individuals who are paraplegic or quadriplegic, regrowing some of the motor neurons so that they can regain some movement. That's the kind of research that is involving these neural type stem cells. So here we have an example of using, again, fluorescently tagged proteins to identify different kind of cells. And of course, you can't do this kind of research in humans because you have to look at the brain tissue after you prepare it. And so this is usually mice or rats, mostly mice, sometimes rats. Because rats are a little bit larger, so depending on what you're doing, sometimes rats are easier to work with. So what they've done here is show that there are new neurons developing in an adult mouse, and they can tell that because there are proteins that a new neuron will express that they have tagged with a green fluorescent tag that an old neuron wouldn't express. So they can tell which are the new neurons and which are the older neurons. So the new neurons are green, the old neurons are red. And they can tell that whatever treatment they've done on this mouse brain, they've gotten some new neurons to form. So that's one of the ways they can tell and test whether they're actually being successful creating or stimulating these stem cells to divide and create new neurons in the brain. So initially, during embryonic development, in all vertebrates, and as well in other non-vertebrate chordates, initially the central nervous system forms as a flat tissue in the ectoderm of the embryo, the outside layer, and it rolls up into a hollow tube. And whatever's on the inside, that becomes the nervous system, and the outside stays ectoderm, it will form the skin of the animal, whatever it is. And that neural tube then will differentiate along its length, with most of the anterior end becoming the brain and the rest becoming the spinal cord. And that's what we see here. And this process is called neurulation. And it's called the dorsal nerve cord because the dorsal side for an animal is the back. And it's not as obvious in humans because we stand upright. But and you might think, well, why say dorsal? Because if it's like that in every animal, but it's not. Chordates have a dorsal nerve cord, but arthropods, for example, have a ventral nerve cord. So it's not, and it doesn't form the same way at all. So the interior in an embryonic stage is a hollow tube that's filled with fluid, and actually that hollow tube filled with fluid continues into the brain. We end up with a canal in the spinal cord and ventricles in the brain that are filled with fluid that tends to flow from the brain down to the spinal cord. And that helps get rid of waste products, contaminants, carries hormones away, and is an important part. The cerebrospinal fluid is a very important part of the nervous system. If you have low amount of cerebrospinal fluid or excess cerebrospinal fluid, that's bad generally for the brain. And so the ventricles in the human brain are pretty small compared to the overall size of the brain because we have a pretty large cerebrum. And you often hear the brain talked about as gray matter and white matter, and it actually looks that way in a freshly dissected brain. If you're going on to anatomy, you might get to dissect a fresh brain from a mouse or a rat. And if you do, you'll see that the gray matter is actually gray, and that's because it's full of neuron cell bodies. And the nucleus and all the mitochondria that are in there give it a kind of a gray color. And the white matter contains a lot of myelinated axons, and so it looks kind of shiny, and it's definitely a different color. And you've probably heard before that not all animal brains are convoluted the way the human brain is. This folding of the surface of the human brain is a way to give it more surface area, because this outside cortex is where all the neurons are. So if you're going to have more neurons, to avoid expanding the brain excessively large, if instead it forms all these folds that give us more cortex without increasing the size of the brain as much. So that's the purpose of that. And I'm going to talk later, I think it's going to be in the second video, about the functions of the different parts of the brain. And most of what we know about how the different parts of the brain function is from individuals who have had damage to those parts of the brain. And these studies have been done way back since the early 1800s. People who've had injuries or strokes and damage a particular part of the brain, correlating that damage with what their deficit was. Was it a speech deficit? Was it a movement deficit? What was it that they couldn't do? That has helped map the brain. In fact, that's how the entire brain has been mapped, is because of accidental injuries or strokes that affected the brain, to see what downstream effect that had on the person's mobility. And sometimes it was post-mortem. A person would have damage to their brain, and they would only find out after that person died which part of the brain had been damaged. Okay, so the central nervous system is where decisions are made, even many unconscious decisions. We like to think of decisions as being all taking place in the conscious part of our brain, but in fact, most decisions are not made in the conscious part of our brain, but there's still being information that's coming in from the periphery is being integrated, and then information is going back out. Commands are coming out of the central nervous system, and most of them we are not conscious of. So brain and spinal cord are the central nervous system. There are nerves going in and out that comprise the peripheral nervous system. And there are groups of neurons where the cell bodies are outside the central nervous system, and those are ganglia, the spinal ganglia, which are on both sides of the spinal cord. Reflexes are an example of an unconscious decision. And many of these reflexes actually take place in the spinal cord. So the knee reflex, or the patellar reflex, is one that's often used as an example. So, and doctors use this to test your nervous system. It's a test of your nervous system. They whack the patellar tendon, and that stretches a muscle, and that sends a signal to your spinal cord. And this little green neuron here, that is an interneuron that integrates sensory information with motor neuron information. So then that's going to send a signal to your muscles to contract, and you're going to move your leg in response. And that is the patellar reflex. Another reflex like that takes place in the spinal cord is the withdrawal from a painful or a hot stimulus. Like if you've ever reached into the microwave to take out a cup of coffee or tea out of the microwave, and it's been much hotter than you expected, and you pick up that cup, your hand will reflexively jerk away from the hot thing, whether it's the handle of a frying pan or a hot teacup, and you'll drop it, and you don't have any control over that. In fact, your muscles contract, and then the conscious part of your brain finds out a split second later. So it happens before the conscious part of your brain even realizes that you've touched something hot, or that your patellar tendon has been stretched. So that is a reflex. And that's why we call it a reflex, because you really don't have any conscious control over that. So in the peripheral nervous system, we have different words for information going towards the central nervous system or coming out of the central nervous system. So afferent neurons are going from the sensory, inputting sensory information to the central nervous system, and efferent neurons are coming from the central nervous system and going to the periphery. And this is mostly motor neurons, but there are a few other types of signals that come out of the central nervous system other than motor neurons. So afferent neurons, this is all of our sensory information, and we're going to talk more about all the different sensory information in the next chapter. Heat sensing, cold sensing, pressure, all that stuff. Taste, smell, vision. We're going to talk about all that in the next chapter. That's all coming in with these afferent neurons into the central nervous system, and sometimes it's only going into the spinal column, and a decision is made without actually going to the brain. It goes to the brain afterwards to tell the brain what has just happened. Efferent neurons are coming out of the central nervous system, and we'll talk about the sympathetic and the parasympathetic nervous system, and also motor neurons. The enteric nervous system, enteric, this is the digestive system. So control of the smooth muscles of the digestive system, that's enteric. And we'll talk a bit about each of these. So the motor system, these are the skeletal muscles, and most of the motor neurons are voluntary. This is under conscious control. The autonomic nervous system, this is smooth muscle. So all of the muscles that surround the blood vessels, the muscles that are part of the digestive tract and the reproductive organs, that's autonomic. And also the speed at which your heart beats, the cardiac muscle. The input from the autonomic nervous system doesn't determine each beat, but it determines how fast the heart will be beating or how slow the heart will be beating. That's under the control of the autonomic nervous system. The enteric nervous system, this is everything to do with digestion, the pancreas, the gallbladder, whether or not the gallbladder is going to empty, the rate at which the pancreas is secreting digestive enzymes into the small intestine. Moving along of food along the intestine by peristalsis and the smooth muscles that do that. That's all the enteric, controlled by the enteric nervous system. So the autonomic nervous system has sympathetic and parasympathetic. The sympathetic nervous system is often called the fight or flight nervous system. This is everything that regulates arousal, energy generation. We've already talked a little bit about this when we talked about adrenaline and cortisol. One of the things that those do is those are produced as a result of the sympathetic division of the nervous system. The parasympathetic is sometimes called rest and digest. So parasympathetic is when you're not in a fight or flight or a high stress state. This is for growth and repair, sleep. That's the parasympathetic. So here are those two. So sympathetic here on the right, parasympathetic on the left. So the parasympathetic, everything, and this isn't a complete list. Neither of these is a complete list, but everything that's on this list are things that are associated with being in a more relaxed state. So pupils, when your pupil of your eye is more relaxed, it's constricted. Salivary glands can secrete saliva. You don't need as much oxygen, so the bronchi and the lungs are constricted. Heartbeat is slowed. Activity of the stomach and the intestines is stimulated. Pancreas is stimulated. Gallbladder is stimulated. Bladder emptying will work. Promotes erection of genitalia. Sympathetic division is things that are associated with fight or flight or stress. dilation of the pupil, or even dilation of the pupil excitement as well, not just bad stress, but good stress. Engagement or excitement as well would fall under this division as well. Dilation of the pupil of the eye inhibits salivary glands, so when you get up to give your presentation to the class and your mouth goes dry, that's the sympathetic nervous system. Relaxes the bronchi and lungs so that you can take deeper breaths, get greater air exchange, more rapid heartbeat. Inhibiting the activity of the stomach and the intestines, and we already talked a little bit about how that is occurring. The muscles that surround the blood vessels that supply the small intestine become constricted, so that limits the blood flow to the intestines, and that's how that works. Inhibits activity of the pancreas, so less digestive enzymes are going to be secreted. Stimulates glucose release from the liver, and inhibits the function of the gallbladder. Those are both the gallbladder, bile is made by the liver. And this is in case, if you're about to run somewhere or, fight somebody, having some extra glucose in the bloodstream might help you survive. So that's why we have that. Stimulates the adrenal medulla. We talked about that already, releasing epinephrine into the bloodstream. This is the rapid response to a stimulus. And this is coming from the brain again. So the brain sees something or senses something that is fearful or exciting, and that stimulates the sympathetic nervous system, and then all these things can result from that. Inhibits emptying of the bladder, promotes ejaculation and vaginal contractions. So kind of two things here that have to do with sex, one from the parasympathetic and one from the sympathetic. So the neurotransmitters, we talked a little bit about those in the last chapter. Neurotransmitters, this is what's being released in the synaptic clefts. So for the sympathetic nervous system, this is usually norepinephrine, which is a very related molecule to epinephrine. And parasympathetic nervous system is mostly acetylcholine, which is the same thing that's the neurotransmitter between motor neurons and muscle cells. Let's talk about the brain a little bit. So when the brain starts to develop, so the neural tube rolls up, different regions of the brain begin to specialize immediately and become visibly different even in an early vertebrate embryo. And we can divide those regions into three big categories and then smaller subdivisions of those. So the forebrain, the midbrain, and the hindbrain, which each have different functions. The forebrain is involved with the sense of smell and also higher functions, the cerebrum. And then the midbrain and the hindbrain, most of the hindbrain and all of the midbrain are part of the brainstem. And then the cerebellum that we'll talk about. Cerebellum is involved with learned motor activities like riding a bike or playing a musical instrument. So later, these three brain structures, they grow at different rates. And in mammals, especially, the forebrain has the most growth. And the forebrain is where we have, that's where the conscious part of our brain is, but not all of it is conscious. The forebrain also regulates sleep, decision-making, emotions. The amygdala is in the forebrain. The midbrain is involved with routing sensory input to the correct part of the brain. So sensory input from the body goes through the midbrain before it goes to the olfactory lobe or other sensory regulatory areas in the brain. The hindbrain is mostly involuntary activities like learned motor activities, the cerebellum, breathing, heart rate, those kind of things are in the hindbrain. Vomiting. So the size of these varies greatly when you compare different vertebrates. And in fact, let me go back one slide here. The hindbrain and the midbrain are often called the reptilian brain because they're so relatively large compared to the forebrain in reptiles, although reptiles have a forebrain. So here's some brains from some different vertebrates. Our jawless fish here, not much of a forebrain. Almost the entire brain here is hindbrain. And that's not because their hindbrain is large, it's because the rest of the brain is small. Sharks as well, not a big difference. Ray fin fish, tiny bit larger for sensory input here, the midbrain, but really not a big change. The same with amphibians. It's not until you get to reptiles and then birds and mammals that you really start to see a big difference. So, and mammals, of course, have the largest brains relative to body size, with certain birds overlapping with that category. Although birds, their organization of their brain is very different, which isn't a surprise considering they evolved their large brains completely separately from mammals. So we have two very intelligent vertebrates that both reach that point along separate evolutionary tracks. So we would expect the organization of the brain to have some differences, and it certainly does between birds and mammals. So there are a few trends, and one trend that we can see among vertebrates is the size of the forebrain increasing. And the forebrain is involved with processing sensory information and making decisions. So certainly that makes sense according to what has happened to vertebrates during the course of evolution. With birds and mammals having the largest brains, about 10 times the size of a fish brain relative to body size, with humans having the largest brain relative to body size, but also other very large-brained mammals would include all the primates and also dolphins. So our brain has about 100 billion neurons and then more than 100 billion glial cells, because there are more than one glial cell for each neuron. And each neuron can have hundreds of connections to other neurons. And it's this anterior neural tube that gives rise to the brain. One thing about the brain, one myth about the brain that started in the 1970s, I don't know where it came from, but it's not true, but I'm certain that all of you have heard it. And that is the myth that everyone uses only 10% of their brain, and what would happen if we use the other 90% of your brain? You know, what would we be capable of doing? And it is not true. Everyone uses all of their brain. It's just completely ridiculous to say that people only use 10% of their brain. It makes no sense, and it's not true either. As we can tell from stroke patients, when somebody has a stroke, it affects them. There really isn't any part of the brain that's not used, that it's okay to take out or have a stroke in, and it has no effect. So the midbrain is part of the hindbrain form what we call the brainstem. So if we look here, here's the spinal cord, and then we have the brainstem. So the brainstem is really all of the midbrain and the hindbrain except for the cerebellum. The cerebellum is this large outgrowth, which is larger in animals that have complicated movements. Like if you compare the way what our brain has to control movement-wise compared to what a fish has to control that doesn't have any limbs, that's why we have such a large cerebellum, and all land vertebrates have a fairly large cerebellum because it's controlling those learned walking and climbing or flying, even movements if we're talking about birds or bats. So that's the cerebellum. So if you learn how to do any kind of a repetitive motor activity like riding a bike or playing the piano, that knowledge resides in your cerebellum. And people who have injury to their cerebellum that's what they lose. They lose those learned repetitive motions. Anything, if you practice it and you get better at it, that kind of a motor activity, what's learning is your cerebellum. Even driving a car, your cerebellum is learning how to drive the car. Your brain is deciding where to go, but how hard to push on the brake, how hard to push on the gas, how much to turn the steering wheel, that's your cerebellum learning that. The forebrain, we divide into two parts, the diencephalon and the telencephalon. The telencephalon becomes the cerebrum, and the diencephalon is everything else in the forebrain. So these are the embryonic brain regions. I am not going to ask you for these on the exam. Telencephalon, diencephalon, don't worry about memorizing these. I'm showing it to you because if I just skip it, it's an obvious thing in the book that I've skipped. So forebrain, midbrain, hindbrain, yes, you need to know that, and what those ultimately form, but not these intermediate embryonic terms. I'm not going to ask you for those. Okay, so the forebrain forms a cerebrum. So the cerebrum is the cerebral cortex and the basal nuclei, which are part of our coordinated movement ability, is the basal nuclei, and the cerebral cortex is all of your thoughts and speech and all that stuff. The diencephalon is all that unconscious stuff in your brain, in the forebrain. And so the thalamus and the hypothalamus, which we've already talked a little bit about that, and the epithalamus, which that means it's right above the thalamus. The midbrain is part of the brainstem. And then the hindbrain, we have the pons and the medulla oblongata. We're just going to call all three of these brainstem. We're not going to distinguish on the exam what the pons does, what the medulla oblongata does. I'm not going to ask you questions to distinguish between those two, but you should know in general what the brainstem does and what the cerebellum does as a part of that. So, okay, so here's our little embryo where you very early in embryonic development for every vertebrate. You can see these three brain regions very clearly. They start to fold and bend and specialize, and eventually the telencephalon and the diencephalon are going to end up on top of the rest of the brain as they expand, especially in mammals. The telencephalon is going to get huge, and it's going to get so large it's going to cover all the rest of the brain. But don't worry about these embryonic names here, the mesencephalon, metencephalon, myelencephalon. I'm not going to ask you about those on the test. So here we go, jumping later. So we have the cerebrum, which is where all of our voluntary motor activities are, speech, our visual cortex, all that stuff is in the cerebrum. And then the diencephalon is the thalamus, hypothalamus, epithalamus. And we see here the little thing that's colored pink here. This is the pituitary gland. And remember, it's the posterior pituitary that's part of the brain. That's why it's colored pink here. And the anterior pituitary is white because it is not part of the brain. It's right up against the posterior pituitary, but they actually form from separate embryonic tissues and come to stick next to each other during embryonic development. So the hypothalamus and the posterior pituitary here are part of the brain, and they develop from the diencephalon. And then we have the brainstem, midbrain and part of the hindbrain, and then the cerebellum, which is also part of the hindbrain. So the cerebrum controls all of our voluntary movements, skeletal muscle. This is also learning, the emotional centers of the brain, the amygdala, memory, the hippocampus, which is in the hippocampus, our perception of the world, where our conscious thoughts are. This is all in the cerebrum. The outside is called the cerebral cortex, and we can map the cerebral cortex, and I'll show you shortly a map of that. I think that'll be in the next video. The two halves of the cerebrum are connected by a thick band of axons called the corpus callosum, and this allows the left and the right cerebral cortices to communicate. And there are, there's so many interesting experiments where patients have had to have the corpus callosum cut because of severe epilepsy. And so now, although they feel and think mostly like a normal person, the two sides of their brain can no longer communicate with each other. So they can show with various experiments that the two sides of the brain have separate thoughts. And I think I have a video in a later chapter to show you about that. That's pretty interesting. And interestingly, in women, the corpus callosum is thicker and contains more axons than in males generally. And we don't know what effect that has, if any. The cerebellum is all about coordinated movements and balance and learned and remembered motor skills, riding a bike, driving a car, playing an instrument, all that stuff. The diencephalon gives rise to the unconscious part of the forebrain. So the thalamus and the hypothalamus, the pineal gland, the posterior pituitary, the epithalamus, and we already talked quite a bit about the hypothalamus and some of the things that controls, including the body's thermostat, because that's where a fever originates. Your biological clock, that originates in the hypothalamus as well. The brainstem is where many unconscious bodily functions are controlled, like breathing and heart rate. The midbrain integrates sensory information, so things coming in from the body, sensory information coming in from the body goes through the midbrain to the various regions of the cerebrum where that information is going to be processed. The pons and the medulla oblongata, this is also a transfer center. Information goes through there to the spinal cord, from the spinal cord to various other parts of the brain. We're just going to call them together part of the brainstem. So the pons and the medulla oblongata are the brainstem. I'm not going to ask you to differentiate between those two on the exam. So the brainstem has several automatic functions there: breathing, heart and blood vessel activity. So that means vasoconstriction and vasodilation, which also means blood pressure. Swallowing, vomiting, regulation of digestion. So all important functions. And here's what the brain looks like from behind. So you can see how large the cerebellum is relative to the rest of the size of the brain. And the basal nuclei, which are, so when you hear that term nuclei are what we call ganglia in the brain. So a ganglion is a cluster of neuronal cell bodies, and we have those outside of the central nervous system in the spinal ganglia, and they're also present in the brain in a few regions. And one group of these are the basal nuclei. So it's groups of neuronal cell bodies that are, it creates a dark area in the brain that is visible to the early anatomists who were mapping regions of the brain. So that's what those basal nuclei are, and they are involved with coordination. So if you're trying to do two things at once, walk and chew gum, basal nuclei are going to help you with that. And in the next video, we'll talk about lateralization of function in the brain, the speech area, visual cortex, all the different parts of the brain and what they do. So the cerebrum controls all of our voluntary movements, skeletal muscle, This is also learning the emotional centers of the brain, the amygdala, memory, the hippocampus, which is in the hippocampus, our perception of the world, where our conscious thoughts are. This is all in the cerebrum. The outside is called the cerebral cortex, and we can map the cerebral cortex, and I'll show you shortly a map of that. I think that'll be in the next video. The two halves of the cerebrum are connected by a thick band of axons called the corpus callosum, and this allows the left and the right cerebral cortices to communicate. And there are, there's so many interesting experiments where patients have had to have the corpus callosum cut because of severe epilepsy. And so now, although they feel and think mostly like a normal person, the two sides of their brain can no longer communicate with each other. So they can show with various experiments that the two sides of the brain have separate thoughts. And I think I have a video in a later chapter to show you about that. That's pretty interesting. And interestingly, in women, the corpus callosum is thicker and contains more axons than in males generally. And we don't know what effect that has, if any. The cerebellum is all about coordinated movements and balance and learned and remembered motor skills, riding a bike, driving a car, playing an instrument, all that stuff. The diencephalon gives rise to the unconscious part of the forebrain. So the thalamus and the hypothalamus, the pineal gland, the posterior pituitary, the epithalamus, and we already talked quite a bit about the hypothalamus and some of the things that controls, including the body's thermostat, because that's where a fever originates, your biological clock, that originates in the hypothalamus as well. The brainstem is where many unconscious bodily functions are controlled, like breathing and heart rate. The midbrain integrates sensory information, so things coming in from the body, sensory information coming in from the body goes through the midbrain to the various regions of the cerebrum where that information is going to be processed. The pons and the medulla oblongata, this is also a transfer center. Information goes through there to the spinal cord, from the spinal cord to various other parts of the brain. We're just going to call them together part of the brainstem. So the pons and the medulla oblongata are the brainstem. I'm not going to ask you to differentiate between those two on the exam. So the brainstem has several automatic functions there: breathing, heart and blood vessel activity. So that means vasoconstriction and vasodilation, which also means blood pressure. Swallowing, vomiting, regulation of digestion. So all important functions. And here's what the brain looks like from behind. So you can see how large the cerebellum is relative to the rest of the size of the brain. And the basal nuclei, which are, so when you hear that term nuclei are what we call ganglia in the brain. So a ganglion is a cluster of neuronal cell bodies, and we have those outside of the central nervous system in the spinal ganglia, and they're also present in the brain in a few regions. And one group of these are the basal nuclei. So it's groups of neuronal cell bodies that are, it creates a dark area in the brain that is visible to the early anatomists who were mapping regions of the brain. So that's what those basal nuclei are, and they are involved with coordination. So if you're trying to do two things at once, walk and chew gum, basal nuclei are going to help you with that. And in the next video, we'll talk about lateralization of function in the brain, the speech area, visual cortex, all the different parts of the brain and what they do.